Understanding adjuvant mode of action in vaccines. The innate immune system is the first line of defense against infection. Innate cells sense the threat of infection via receptors that recognize immune defense triggers, like lipopolysaccharide, or viral DNA, or stress signals from body cells. Activated innate cells then secrete immune mediators. The secreted immune mediators trigger an inflammatory response and attract more innate cells from the blood. Some innate cells directly combat pathogens. Other activated antigen-presenting cells, such as dendritic cells, break down and present pathogen antigens while migrating to the lymph node, where they trigger an adaptive immune response. The antigen-presenting cell is the bridge between innate and adaptive immunity. Vaccines containing purified antigens may have limited or no immune defense triggers, essential for generation of the innate response. Such a vaccine would elicit a mild innate immune response. Injection of a vaccine containing purified antigen and an adjuvant, such as alum or oil in water emulsion, enhances innate cell recruitment at the injection site. The role of the adjuvant is to mimic natural immune defense triggers and help elicit transient activation of the innate immune response. This may cause visible local inflammation. A vaccine containing a purified antigen without adjuvant elicits mild activation of antigen-presenting cells, such as dendritic cells. These cells then break down and present antigen as fragments or peptides on their surfaces. A few dendritic cells migrate to the draining lymph node. A mild innate immune response may be insufficient to trigger effective adaptive immunity. Vaccines containing antigens and adjuvant elicit highly activated dendritic cells. Antigen-presenting cells migrate in large numbers to the draining lymph node. Appropriate activation of antigen-presenting cells by antigens and adjuvant is key for long-lasting immunity. The dendritic cell can be highly activated by adjuvant binding to toll-like receptors, for example. The cell internalizes antigens, breaks them down, and presents large quantities of antigenic peptide complexed with HLA class II at the cell's surface, next to large numbers of co-stimulatory molecules. The HLA class II peptide complexes are now visible to CD4-positive T cells. Some adjuvants may stimulate innate cells and enhance innate immunity in other ways. Activated antigen-presenting cells bridge the innate and adaptive immune response by migrating to the T cell zone of the draining lymph node and interacting with T cells. T cells recognize and bind to antigen-presenting cells. The activated dendritic cell secretes immune mediators called cytokines that stimulate T cell division and the differentiation of memory cells. The immune profile of the adaptive response is influenced by adjuvanted vaccines and may result in a greater diversity of CD4-positive T cells. Activated T cells secrete cytokines to stimulate B cells to divide into antibody-secreting plasma cells and memory B cells. Immune cells and antibodies with a broader specificity leave the lymph node and enter the bloodstream to fight infectious diseases. Greater understanding of the role of the innate response in eliciting specific adaptive immunity has led to an increased knowledge of the role of adjuvants in vaccine formulation. Adjuvants enhance and modulate the immune response to antigens by mimicking natural immune defense triggers. The selection of particular antigen-adjuvant combinations in the design of vaccines can help to address remaining challenges linked to complex diseases and specific populations. Adjuvants can be key contributors in developing efficacious vaccines aiming to deliver enhanced and sustained protection.